Welcome, everybody. Bienvenue à tous et à toutes. My name is Amy Ryan. I'm an Associate Dean for Graduate and Postdoctoral Studies in the Faculty of Medicine and Health Sciences at McGill. And thank you so much for joining us today. I want to begin by acknowledging that McGill is located on land which has long served as a site of meeting and exchange amongst Indigenous peoples, including the Haudenosaunee and the Anishinaabe nations. McGill honors, recognizes, and respects these nations as the traditional stewards of the lands and waters on which we meet today. And I invite everybody to explore this further. There are links on our webpage as well as others to understand further these Indigenous peoples and their care for the land. We are here thanks to the generosity and vision of the Cole Foundation. Nous sommes aussi aujourd'hui grâce à la générosité et à la vision de la Fondation Cole. The Cole Foundation was established in 1980 by Jack Cole. Jack had lost his daughter Penny to leukemia on her 20th birthday, and the foundation established the Jack Cole Chair in Pediatric Oncology and Hematology at McGill, as well as the Penny Cole Laboratory at the Montreal Children's Hospital. Since Jack's death in 2004, the foundation has continued to carry on this mission with a renewed focus on research and childcare for young patients affected with leukemia and leukemia-related disease. Over the past 15 years or so, the foundation has offered fellowships to clinical, doctoral, and postdoctoral scientists dedicated to research in these leukemia and leukemia-related diseases in children and in young adults. The continued support of the Cole Foundation enables our early career scientists to be at the forefront of pediatric leukemic research. Not only does the Cole Foundation program give recipients the means to focus on their research, but it also provides them with a really important opportunity to network with others who share their interest in pediatric cancers. And we know that these networking opportunities are so important for our developing scientists. This year, we've had to be a little bit more creative about how we do that, and by taking the Cole Foundation Research Celebration Days online. This is the second part of a two-day celebration. Last week, many of you may have joined us, and we heard from several researchers whose work has been made possible by the Cole Foundation. And if you missed their talk, New Voices, New Ideas, we can share that video with all of you who have registered today so that you have an opportunity to listen to these individuals speak, and as well as our keynote speaker today, Dr. Scott Armstrong. We are tremendously grateful to Jack's nephew, Barry Cole, currently president and CEO of the Cole Foundation, for his steadfast and continued support. But first, before we go to our keynote speaker, I would like to introduce our moderator, Dr. Janusz Rak, who is the Jack Cole Chair in Pediatric Hematology-Oncology and professor in the Department of Pediatrics at McGill University. Dr. Rak, I'll hand it over to you now.
Well, thank you, Amy. This is a very kind introduction, a very warm welcome to everybody, which I'd like to join. Good afternoon, everyone. It is a considerable pleasure and honor for me to introduce today's keynote speaker, Dr. Scott Armstrong. For those of us who produce pediatric oncology literature, Dr. Armstrong perhaps doesn't need any introduction, but please let me make just a few comments to highlight his truly inspiring career and transformational contributions. Dr. Armstrong received his medical training and PhD from the University of Texas Southwestern Medical School and completed several high-profile fellowships, not the least of which was at Dana-Farber Cancer Center in Boston with late Dr. Stanley Korsmeyer, the pioneer in the field of apoptosis lymphoma research and immunology. Dr. Armstrong's subsequent stellar career led him to his present prestigious leadership appointment as chair of the Department of Pediatric Oncology at the Dana-Farber Cancer Institute, and David Nathan, a professor of pediatrics at Boston Children's Hospital at Harvard Medical School. But of course, Dr. Armstrong's prominence is also reflected in a number of other accolades, of which I'd like to mention just a few, not to occupy the whole hour that is allotted to this event. So among them, there's Paul Marks Prize for Cancer Research, E. Johnson Award from the Society of Pediatric Research, the Meshek Prize for the American Society of Hematology, membership in the National Academy of Medicine, chairmanship of program committees with the American Association for Cancer Research, where he also served as on the board of directors, and many, many others. Dr. Armstrong's publication records is truly enviable, which is among scientists a good thing. This record comprises, among others, over 120 peer-reviewed publications, which only within 5 years, as I calculated, included 2 papers in Science, 8 papers in Nature journals, and 4 papers in Cell and Cancer Cell, which is pretty remarkable. Other 43 scholarly chapters and reviews that he published. But above all, Dr. Armstrong is a pioneer, a true pioneer in the field of pediatric leukemia, where he paved the way to new understanding and treatment of these perplexing diseases in which so much still needs to be done. For example, he made seminal contributions to elucidating oncogenic pathways driving progression of acute leukemias in children, including pivotal roles of the MLL gene rearrangement in acute leukemias. Perhaps the impact of the gene on cellular chromatin and stemness led Dr. Armstrong to his current interest in discovery and targeting of genetic as well as epigenetic drivers of pediatric leukemia progression, and hence the title of his talk, Targeting Chromatin Complexes as Therapeutic Opportunities in Cancer. Dr. Armstrong also leads important basic and clinical studies on drug resistance, including development of new agents, some of which have already entered clinical trials. We could spend another hour talking about Dr. Armstrong's research and influence, but perhaps we should hear about this from himself. And I'm very honored to be in a position to participate in this event. For those of you who have questions, I would like to suggest that please direct these questions to the email address specialevent.med@mcgill.ca, and they will be relayed to us during the presentation. And on that note, Dr. Armstrong, thank you for joining us today at this virtual event, and the virtual floor is yours. Thank you.
Thank you, Dr. Rack, for that very kind introduction. And I also would like to personally thank Mr. Cole for his support and the Cole Family Foundation for their support for pediatric leukemia and pediatric cancer research. It's incredibly important to have supporters like you to help us do what we do. So I'm going to share my screen now. Here we go. So as was mentioned, I'm going to talk about some of the work that we've been doing focused on understanding how specific oncoproteins drive the development of both pediatric and adult leukemias, and how we might use that information to develop new small molecules that disrupt those mechanisms for therapeutic opportunities. And I'll kind of go from the beginning to the end, from the kind of basic biology to some of the new small molecules in the talk. I do have a few disclosures. These are shown here. Quite a bit of drug discovery going on in this space. The only thing relevant for today is Syndax Pharmaceuticals pays us to do some preclinical work for them, and I'm going to talk about one of their compounds. So my lab is interested fundamentally in 2 or 3 questions, all of which are related to the hematopoietic system that is shown here on the left. And I suspect this group probably doesn't need too much introduction into the hematopoietic hierarchy, but just to remind you that the long-term hematopoietic stem cell is the only cell in hematopoietic development that can both divide to make copies of itself and also give rise to all of the potential blood cells shown down here, that process called self-renewal. And as hematopoietic development proceeds, a number of steps down the differentiation pathway happen until you have a fully developed blood cell of a specific type. And of course, we believe, and many studies have shown, that disruption or corruption of this process is a central part of leukemia development. We, as was mentioned, are focused on this particular chromatin complex, the MLL complex, and I'll tell you why in just a minute. But both leukemias driven by mutations in that complex and also other types of leukemias that we think are taking advantage of that complex to drive aberrant leukemogenic gene expression. So I became interested in the question as to how translocations of the MLL gene on chromosome 11 Q23 drive leukemia from experiences I had during my pediatric hem-onc fellowship in the late 90s, when I was introduced to the problem, particularly in infants that have rearrangement of chromosome 11, Q23, they do particularly poorly, and really have now spent the last 20 years trying to understand how we might do something about that. And just a quick background, in the late 70s, here the first picture of a 11q23 translocation, where a part of chromosome 4 shown here is stuck onto chromosome 11 shown here, and that encodes for what's now the famous 411 translocation. That is one of the most common translocations involving 11q23. And then in the late 70s, it was shown that this is a recurrent abnormality in various leukemias associated with infants. That is, if a child less than 1 comes to the clinic in Montreal, in the United States, wherever, over 70% of those leukemias are going to have rearrangement of the gene that was in the gene on chromosome 11q23. It's also these translocations found in about 30% of pediatric AMLs, the infant leukemias most often being acute lymphoblastic leukemia. And then in the late 90s, the breakpoint, or sorry, in the early 90s, the breakpoint on 11q23 was cloned by a number of groups and found to be the MLL gene that has a number of other names shown here. I'll refer to it as MLL1 throughout the talk. Then in the mid-90s, Stan Korsmeyer, my postdoctoral mentor, as Dr. Ack mentioned, demonstrated that the wild-type MLL1 is a homolog, functional homolog of the Drosophila trithorax gene that was known to control the expression of developmental transcription factors, those the HOX cluster, the homeotic transcription factors, and another transcription factor called MEIS1 that heterodimerizes with HOX proteins to influence development in many different tissue types, including blood. That relationship is important because I'll talk about these genes as relevant targets in leukemia throughout the rest of the talk. And then in the late 90s, Mike Cleary's group at Stanford and Terry Rabbitt's group in England demonstrated that the fusion protein, the protein product of the MLL translocation, is indeed oncogenic, and developed mouse models, one via retroviral mediated transduction of blood cells, and one as a genetically engineered mouse model, but both show that the fusion protein is leukemogenic. And I would say the development of those two models really set the stage for everything that's come and many of the discoveries that I'm going to tell you about today. So this is a schematic of the MLL protein shown here. It's a very large nuclear DNA binding protein. And when the translocation occurs, the N-terminus of MLL shown here is transcribed in frame or fused in frame to the C-terminus of what's some 70 or 80 different translocation partners. So this is invariant, and the translocation partner does vary. However, there are a few translocations that are particularly common, and this is a pie chart showing the different potential fusion partners, with AF4 of the 411 translocation or AF9 of the 911 translocation being the two most common. And you can see these four genes here, AF4, AF9, ENL, and AF10, really account for the majority. That's relevant because I'm going to tell you about the proteins encoded by those genes in a little bit later. So in the early 2000s, when I started as a fellow, really that's where we were. We knew the fusion protein was oncogenic. We knew that the fusion protein probably bound to DNA. We didn't know much more than that. And then quite an important breakthrough by David Alice's group at Rockefeller University and Eli Kanani's group in Israel demonstrated that the wild-type MLL protein has enzymatic function in that this C-terminal set domain modifies histone H3 on lysine 4, puts 3 methyl groups on that residue, and subsequently, as most of you probably know, that modification has been shown to be associated with active gene expression. It has also subsequently been shown that the wild-type MLL is important to maintain appropriate developmental gene expression, like the HOX and MEIS genes, but others as well. Much work has been done in the biochemistry of the MLL complex. This is the wild-type MLL complex, and you can see there are a number of associated proteins that have various functions. One I'm going to talk about a fair amount today is this interaction here between the N-terminus of MLL and this protein called menin. Also, via microarray approaches and now RNA-seq, we know a lot about the gene expression program of MLL rearranged leukemias. This was some of the early work that I did as a fellow, and showing that the HOX genes here at HOXA9, one of the HOXA cluster genes, are highly expressed in MLL rearranged leukemias. Each one of these bars is a leukemia, whereas in ALL at least, the HOX genes are not expressed unless the MLL translocation is present, suggesting that whatever is happening with the fusion is likely a gain of function or is somehow positively influencing HOX gene expression. So over the next 10 years or so, to get everyone up to speed, we and a number of other groups demonstrated that when you introduce the MLL fusion, in this case via retroviral mediated transduction, into either hematopoietic stem cells or downstream myeloid progenitors, you can induce leukemia from either of those cells of origin. And the leukemia stem cell in these models always looks like a myeloid progenitor that's inappropriately expressing high levels of stem cell associated genes, like the HOX genes, like MEIS1, and other stem cell programs like the Wnt beta-catenin pathway, such that the leukemia really is a hybrid between, or the leukemia stem cell is a hybrid between a normal progenitor cell and expressing many components of stem cell-like gene expression programs. So that obviously leads to the question as to how does the MLL fusion do this? How does it drive gene expression in the wrong cell type? And can we use that information to do anything about the leukemia? So as this was happening, the biochemistry of the chromatin complexes that are important here were being defined in quite a bit of detail. And I've already mentioned to you the MLL complex shown here. Two other relevant complexes are the super elongation complex shown here, where the central enzymatic component of that complex is CDK9, which phosphorylates RNA polymerase II to allow RNA polymerase to transcribe genes, actively transcribe genes. And the .1L complex, .1 being the enzymatic component, which puts 2 or 3 methyl groups on histone H3 lysine 79, that modification, much like H3K4, also associated with active gene expression. It's obvious why this complex is important. It may not be immediately obvious to you why these two are, but if you remember, I mentioned AF9, AF4, AF10, and ENL, which is similar to ELL, are recurrent and common MLL fusion partners. So the current thought is that the MLL fusion protein essentially locks together these complexes, MLL and superelongation complex, or MLL and .1, in a way that's aberrant. And that is why gene expression cannot be shut off as it should be. And that's what leads to the development and continued proliferation of leukemia cells. So needless to say, that's prompted us and others to try to understand which of the components can be targeted with small molecules. And when you do that, what happens to leukemia and what happens to normal cells. And this is a list of potential targets against which small molecules have already been made. .1, the enzyme here, CDK9, studies have been ongoing with trying to inhibit that kinase for quite some time. And then a number of protein-protein interactions here, MLL-menin, BRD4, which is also in some of these complexes bound to acetyl-lysine, and WDR5, which is a part of the MLL complex. So a lot going on here, still a lot of work to be done, but I'm going to take you through quickly a little bit of work on .1 and then more detail on MLL-menin. So with that background in mind, we were interested in .1 mostly because, well, 2 reasons. One, it was an enzyme against which a small molecule could be potentially developed. And it was at the time new and not too much was known about the modification that the .1 enzyme placed, this H3K79 methylation. So we made a conditional knockout mouse, Catherine Berndt, a pediatric chemo fellow in the lab at the time, transformed mouse cells with MLF9 and then inactivated .1. And the important thing, this was published a number of years ago, but the important concept here is that when .1 was inactivated genetically, you saw relatively specific changes in gene expression. That is, the MLL fusion-driven program that we know, based on other studies, is really the direct targets of the MLL fusion protein. Those genes were particularly sensitive to .1 loss whereas most genes did not change expression, at least in the short to midterm. That was important because these histone modifications tend to be associated with every or most actively transcribed genes. So if there wasn't some type of selected dependency for gene expression, then therapeutically there would be presumably no potential therapeutic index. And this now has played out in many different ways and many different complexes, that and is a fundamental finding in the field of chromatin biology, is that even though these complexes can be associated with thousands, if not tens of thousands of genes, when you inactivate one or two components of those complexes, you tend to get very specific changes in gene expression. So for a therapeutic purpose, that is obviously important. So just to say that this moved all the way from development of a small molecule shown here bound to the crystal structure of .1L. And we were able to use that to show that what K79 methylation was doing was preventing repression of these genes that should be shut off at this stage in hematopoietic development, like the HOX and MEIS genes. And that the .1L inhibitor inhibited the proliferation of MLL rearranged leukemias, as we might expect based on the gene expression, and another subtype of AML that I'll get into in a minute. The phase 1 trial for this drug actually has been completed. Remarkably, there was no dose-limiting toxicity, and there was biological, and in two of the cases, significant clinical activity, and 2 patients went into complete remission with this small molecule. So proof of principle that 1, it's not particularly toxic to do this, and 2, you can get biological and clinical activity. But for this small molecule, leukemias appear to be relatively adept at figuring out how to get around it. That is, the responses only last for a month or 2, and then the leukemias become insensitive to .1 inhibition. We're continuing to study that. We can recapitulate that in model systems. So that continues, but in the meantime, we switched our thoughts or added to our thoughts in targeting this interaction here, the interaction between MLL and menin. As I mentioned earlier, Mike Cleary's group that had developed the MLL translocation models had shown that this interaction is important for MLL-driven leukemia. And this is the crystal structure here with a small molecule that we've recently developed with the company Syndax bound to this pocket here in menin, to which these 5 amino acids on the N-terminus of MLL, 5 amino acids here, this being the N-terminal methionine, bind to this pocket. And that interaction is critical for continued transformation of MLL fusion-driven leukemia. So the concept is if you can disrupt this interaction with a small molecule, perhaps you can get therapeutic benefit. And I should point out that the initial probe compounds for this approach were developed by Jolanta Rebecca's group, the University of Michigan. So this is some data from that small molecule where we treated a number of different leukemia cell lines, some of which are shown here, with MLL rearrangements, one of which does not have an MLL rearrangement. And you can see the IC50 for proliferation is quite low, in the low nanomolar range for the MLL rearranged lines. In general, the non-MLL rearranged lines are insensitive to inhibition of proliferation. I'll tell you the exception to that rule in just a moment. This on the right here is a glycerol gradient, which some of you may know basically separates proteins based on the size of either the protein or the complex that they're in. And you can see menin resides in some large megadalton-sized complexes. When you treat with the menin inhibitor biochemically, menin then it comes out of those complexes and runs as the free protein. And if you look at gene expression shown here on the bottom, some of the genes that, again, we know are important MLL targets, MEIS1, PBX3, MEF2C, these are all transcription factors that we've previously shown to be important in this disease. Those genes are getting suppressed at day 2 and at day 7 after treatment with the small molecule. So it does pretty well what we think that or hypothesize this inhibitor should do. And then if we look at chromatin, this is really, to cut to the chase on the MLL part, probably the most interesting biological finding from this is that when you disrupt the MLL-menin interaction, menin comes off of chromatin. This is ChIP-seq for menin, and most of you probably know this technology, and you can see you lose the menin signal on chromatin. MLL itself, which interacts with menin, actually mostly across the genome is not lost. MLL is still associated with chromatin broadly. .1 actually is destabilized and lost from chromatin. That's an interesting aside that we don't quite understand the mechanism for that. But if you look here at specific loci and focus on menin, it's lost across the loci, be the HOXA cluster or some of these other transcription factors. But MLL itself is not lost at the HOX cluster, but is lost here at MEIS1, MEF2C, JMJD1C, a number of other loci. So what's this telling us? It's telling us the disruption of this interaction leads to specific loss of MLL occupancy at certain loci in the genome. And it's those loci that get shut off, the gene expression. So we think this is part of the reason there may be a therapeutic index here is because we're not stripping all of MLL off chromatin. That probably would be bad and toxic to hematopoietic cells, but we're getting relatively specific changes in chromatin state and gene expression. This molecule has very good pharmacokinetic properties and drug-like properties. So we were able to do a number of in vivo experiments, both with pure mouse models of leukemia and with human leukemias engrafted in immunodeficient mice, shown here. We can put the drug in the food of the mice, makes these experiments much easier for those doing the experiments. And here's the response, a survival response of an MLAF9 mouse model. For those of you who know this mouse model, you know these mice die very rapidly, less than 60 days, they'll succumb to leukemia. But if we add the food that has the VTP-50469, the menin inhibitor, during this window, we actually eradicate the leukemia and the mice ultimately survive to 2 years, which is about a mouse's lifespan. So this is a really dramatic response in this model, both because the inhibitor works, we think, via the mechanism we hypothesized, but also it just has very good pharmacokinetic properties and where we can get tremendously good inhibition of the interaction. We see also, I'm going to skip this just to get to the next part, dramatic responses in human leukemias growing in immunodeficient mice as well. And in fact, we're able to eradicate some MLL rearranged ALLs from PDX or from immunodeficient mice. So again, a very impressive response. So to summarize what we think is happening here with menin inhibition versus .1 inhibition in MLL rearranged leukemias, if this is what the steady state, if you will, in the leukemia, we inhibit with an enzymatic inhibitor .1, we get some initial response, but ultimately the leukemias figure out how to deal with that. I'll point out that the complexes largely remain on chromatin when we do that. And we think that may be part of the reason that the enzymatic inhibition is just not sufficient. But when we disrupt this protein-protein interaction, menin comes off chromatin, MLL comes off, and actually, I didn't show you this, but menin and MLL are destabilized as a result of that loss. And so we think disruption of the complex is a more dramatic thing to do to the cell. And actually, the gene expression changes are much more rapid, and therefore, hopefully, the response less likely to, or the cells less likely to develop resistance. So we continue that work on MLL rearranged leukemia. I'll tell you a little bit about the clinical trial at the end. But we began a few years ago to wonder if the mechanisms that are being used in MLL rearranged leukemias that represent about 10% of all leukemias, as some of you may know, might be also being taken advantage of in more common types of both of adult AML, but AMLs that are also found in pediatrics. And we went to the groups of leukemia to study this, the groups of leukemias that also express HOXA cluster or HOXB or HOX genes and MEIS1. So this is gene expression data from Tim Lay's group. And I'm sure you know how to read this. These are the individual genes. Each column is a patient sample. The MLL rearranged samples are here. You see the HOXA cluster and MEIS1. And there's a big chunk of other AMLs, these are all AMLs here, that have expression of the HOX genes and MEIS1. And about half of those have mutations in a protein or a gene called nucleophosmin or NPM1. This mutation is a specific mutation called the NPM1C mutation and is found in about 30% of adult AML. So this is much more common. The biological effect of that mutation, or one of them, is for the NPM1 protein to become cytoplasmic. That's mostly how it's followed. And these mutations usually occur with other mutations, like mutations in DNA methyltransferase 3A. So Michael Kuhn, a postdoctoral fellow a few years ago, decided to look to see if MLL, in this case, the wild type MLL, might be important for continued proliferation of these leukemias using both mouse models and human cell lines. He did a number of studies with a number of CRISPR screens, one of which is shown here, where he was developing guides to various domains of the MLL protein, MLL gene. And what he found is that the part of the gene that encoded for the MLL-menin interaction, cells were particularly sensitive to targeting that region of the gene. And he went on with the probe compounds to show that MLL-menin interaction, here, presumably the wild type MLL-menin, led to differentiation and ultimately death of the NPM1 mutant AML cell lines as well, suggesting that this leukemia may be using similar mechanisms to maintain itself and to continue proliferation. About the same time, a number of groups, one of which I think John Dick has spoken in this session before, showed that NPM1 mutations are most often found in people in cells that look like myeloid progenitors as opposed to stem cells, whereas the DNMT3A mutation is almost always found in hematopoietic stem cells. So this made us wonder if perhaps the NPM1 mutation, is driving leukemogenesis in the setting of a myeloid progenitor, much like the MLL fusion might be. So Hannah Uckelman, a fellow, set up the following experiment with various conditional mouse models, where she had either wild-type mice that just had the DNMT3A mutation, mice with the NPM1 mutation, or the double mutant, and then looked at cells that are myeloid cells that are moving down the developmental pathway. So stem cells, early progenitors, common myeloid, granulocyte macrophage progenitors. This is HOXA9. We know that normally that gene's being shut off during myeloid development. But if you have the NPM1 mutation there, whether or not DNMT3A mutation is there, the HOX genes are inappropriately maintained in a downstream myeloid progenitor, which is very similar to what we see with the MLL fusion. If you do RNA-seq on these cells, you can see the whole HOX A and B cluster are activated in downstream myeloid progenitors inappropriately, again, much as we've seen in the MLL leukemias. And this just shows, looking at more long-term hematopoietic stem cell signatures, showing there's a more extended signature expressed in these progenitors that really shouldn't be expressed, at least at this level. And then she asked a related question, but slightly different. That is in that case, in the experiment I just showed you, the NPM1 mutation was activated either in stem or progenitor cells. We didn't direct which cell type we wanted to activate the mutation in. But in this one, we do it in progenitors, saying, okay, let the cells develop and then get the mutation and see what happens. And we're able to show that in vitro, this is colony replating, that the NPM1 mutation can confer enhanced replating of the granulocyte macrophage progenitors when the mutation's activated there. And this is showing transplantation, so enhanced engraftment. And then ultimately the mice, after a secondary transplant, will develop leukemia independent of the cell in which we activated the mutation. So the myeloid progenitors, in this case, can act as the cell of origin in NPM1 mutant AML. So to summarize this, in relation to what I've shown you about the MLL rearrangement, we think that the DNMT3A mutation is in the stem cell. That's been shown very nicely by John Dick's group and Ron Slush, Robbie Majetti, and others. And that then gives you downstream progenitors that have these mutations, and the NPM1 mutation is then acquired in a myeloid progenitor, leading to aberrant proliferation and activation of these HOXA, B, and MEIS1 genes that I've talked about in the setting of the MLL fusion. And you can probably tell where I'm going here. This really suggests that perhaps the menin inhibitor and other mechanisms that we're using to target MLL fusion leukemias might be relevant in this disease. So Hannah then took the small molecule MLL menin inhibitor and put it on OCI-AML3 cells. This is a human cell line with an NPM1 mutation. And you can see at day 6, a low to mid nanomolar IC50 for inhibition of proliferation. Mouse leukemia cells growing as colonies shown here are quite sensitive to the menin inhibitor. And the primary phenotype, as is the case with MLL, is differentiation of the cells and then ultimately death. If you look at gene expression, you see interesting changes in gene expression with the MLL menin inhibitor shown here. So this is MEIS1, again, one of the most sensitive genes to MLL menin inhibition in the setting of an NPM1 mutant AML. PBX3, I think I mentioned that to you earlier in the MLL leukemias as well. The Hox genes decrease expression, but slowly. And we're studying this. We don't understand at the molecular level why it is that we're seeing these differential kinetics in terms of gene expression changes, but the Hox genes will ultimately decrease expression. But again, that's more inconsistent and kinetically much slower than the loss of expression of these genes. And it's not RNA stability. We've actually looked at that. This prompted a set of experiments we needed to do to show that MEIS1 is actually relevant in the setting of this leukemia that had not been done before. So Hannah did a CRISPR knockout of MEIS1, and this is in mouse leukemias, and you can see that suppresses colony formation. And importantly, if we take the MEIS1 gene, express it from a retroviral promoter, so that's no longer sensitive to inhibition of MLL menin, that will rescue the cells from the differentiation and death induced by the inhibitor. So here's the control cells treated with the inhibitor. Here are the cells that were MEIS1s overexpressed, treated with the inhibitor, and you can see they continue to proliferate. So this doesn't mean MEIS1's the only relevant target, but it does mean it's a relevant downstream target that is influenced by the MLL fusion. I'm sorry, the MLL menin inhibitor. So I won't go through this in detail, just other than to say that what we found in the setting of the NPM1 mutant cell lines treated with the menin inhibitor is exactly the same as what we found in the MLL cell lines. That is, specific loci lose MLL occupancy, all loci lose menin occupancy, and the places where you lose MLL occupancy is where you lose expression. So this phenomenon, again, of loci behaving differently, having different dependencies, and the genes where we get rid of the MLL protein are very sensitive to that perturbation. So next, the in vivo experiments for this approach. What does the menin inhibitor have at potential activity in this disease? And Hannah did a couple of different types of experiments here, one of which really is interesting and sets up a number of future experiments. I didn't tell you this in great detail, but what we see is that when we activate the NPM1 mutation in downstream myeloid progenitors, we inject those into mice. Those progenitors will hang around for 6, 8, 12 months, continue to proliferate, but actually not give you leukemia for a long time. So we call these pre-leukemic. We can talk about the nomenclature around that, but these progenitors are sitting there self-renewing for a long period of time. Then if you transplant them to another recipient, they will drive the development of leukemia. So the obvious, perhaps, experiment was to see if we could intervene with the menin inhibitor in this pre-leukemic state and prevent the ultimate progression to leukemia. So one of those experiments is shown here. So this is the engraftment in the secondary recipient mice. You can see it increasing over time, and those mice ultimately die. And if you intervene with the menin inhibitor here, you can eradicate these self-renewing cells, and ultimately those mice then live a normal lifespan. So this is showing that prior to the development of the full-blown leukemia, you can intervene to prevent progression all the way to leukemia. That's interesting and has a number of potential ramifications for future ways of thinking about treating patients that might have a high risk for development of leukemia. But what about now in the setting of fully developed AML? And these are human PDX experiments shown here too. One human leukemia with an NPM1 mutation and a mutation in the FLT3 receptor tyrosine kinase. Some of you know, that's a very aggressive type of adult AML. Kids do get this as well, but more commonly in adults. And here's a more genetically complex human leukemia. And you can see that the MLL menin inhibitor, the treatment period shown here in gray, eradicated the disease in this PDX, and 4 of the 5 mice here were rescued from death from the leukemia by the inhibitor. And you can see we actually, in this case, waited until very late in the disease. Mice were already sick before we started treating them, and we were able to rescue the survival of 4 of 5 mice. So this inhibitor, this approach really does appear in preclinical experiments to have pretty impressive activity in this subtype of AML. So one of the questions that's common that we get whenever I present this is, well, what's the connection between the NPM1 mutation and MLL? I told you the NPM1 mutant protein tends to reside in the cytoplasm, or at least was thought to largely reside in the cytoplasm. And we're just started these experiments, but have some early data that's particularly interesting. And we've used a system that was initially developed by a number of groups, Jay Bradner's being the one that we've worked with most when he was at Dana-Farber. This cell line generated by Peggy Goodell's group from Baylor, where they engineered a human cell line to have a mutant version of FKBP fused to the mutant NPM1C. And this FKBP protein can be targeted with a heterobifunctional small molecule degrader. I'm not going to get into the chemistry of that. Many of you probably know about this, but basically what it allows us to do is to degrade the mutant NPM1 protein in a human leukemia line and look to see what happens right after or days after degradation of the mutant. So here's the mutant protein, and here is after treatment with the small molecule degrader. At 3 hours, you can see it degrades the protein quite quickly. And you can see here proliferation of the cells after degradation of the mutant NPM1. The cells stop proliferating and ultimately die. And here's showing differentiation and apoptosis. That's presumably what you would expect when you degrade an oncogene in a human cell line, and indeed that's the case. So this, as I mentioned, was work published by Peggy Goodell's group. An antibody's recently been developed. This is gene expression, just showing that you lose HOX and MEIS gene expression when you degrade the mutant. So there is some relationship between the mutant NPM1 and the HOX and MEIS genes, which I've shown you in terms of the MLL-menin inhibitor, but this shows it in terms of the oncoprotein. And recently, an antibody to the mutant NPM1 protein that recognizes selectively the NPM1 mutant protein has been developed, with which we can do chromatin immunoprecipitation. And what you see here, and this was shown by another group initially, that there is some, it appears, occupancy of the NPM1 protein on the HOXA9 and the HOXA cluster genes and MEIS1. That is, the mutant NPM1 that's also in the cytoplasm is in the nucleus as well, and localized to chromatin at the genes that we think are being driven by this oncoprotein. And when you degrade it, you lose the signal, importantly, showing that this indeed is a specific signal. And you also lose MLL from chromatin, particularly at MEIS1, but also a decrease at the HOX locus. So here it suggests that the mutant NPM1 protein is somehow maintaining the integrity of the MLL complex on chromatin. I've already told you that's important for these leukemias. So then if you look at the MLL-menin inhibitor, what's it doing to the oncoprotein? And just like I showed you for MLL, the oncoprotein comes off of the MEIS1 locus, off of other loci, but not so much off the HOX loci. And depending on how much MLL you lose, 40%, 50% signal, 60% signal, you actually see something of a dose response here in terms of loss of gene expression. That is, the more MLL you lose after treatment with the MLL-menin inhibitor, the more mutant NPM1 you lose, the more you lose gene expression. So this is showing you there is an intimate relationship between the mutant NPM1 protein and MLL and menin on chromatin, and we're continuing to work through the mechanisms of that. So to summarize this, I've shown you that NPM1C, the mutant oncoprotein degradation, leads to loss of MLL on chromatin. This is showing degradation. And the MLL-menin inhibitor leads to loss of both the NPM1 protein and MLL on chromatin. And we think that it's this relationship that is the reason, or at least one of the reasons, why the NPM1 mutant AML is sensitive. So based on all that, we're starting to think about, and I'm not going to show you any data in this regard, this current thought process here. So some of you, many of you may know that many people have what's called clonal hematopoiesis as they age. That is, they have stem cells that have mutations in genes that we know are important for AML development, like DNMT3A, but they won't necessarily develop AML. A lot of work going on to try to understand who has high risk of development of AML, and we'll probably figure that out in the next 10 years or so. But it's certainly the case if you develop an NPM1 mutation in the setting of clonal hematopoiesis, then you are going to develop AML. And so the concepts that are starting to develop is if we screen and watch patients closely enough, can we think about intervening somewhere in this step of the disease prior to the development of full-blown AML? And plenty of work to be done before we're ready to do that type of clinical trial, but some interesting concepts nonetheless. I'm going to go through this rapidly to say there's another subtype of AML that's particularly aggressive. This is almost only found in pediatrics, and it's pediatric AML with rearrangements of the NUP98 gene. This is a nuclear porin gene that's fused actually to many of the same fusion proteins, or at least proteins involved in the same processes that the MLL fusion the MLL fusion proteins are involved in. And this is extremely aggressive. But we've previously shown that MLL is important for this leukemia in a paper we published a few years ago in Cancer Cell. And now, as you can imagine, are looking at the menin inhibitor. And this is just one PDX experiment done with Andy Kolb, Andy Kolb's group. And this is similar to the ones I've shown you before. Inject the pediatric AML into immunodeficient mice, treat the mice with either control food or the menin inhibitor. And you can see with 2 different concentrations of the menin inhibitor, you get prolonged survival of the mice. And this is just showing the leukemia in the peripheral blood. So pretty dramatic response there as well. So we're continuing to work on this, but the mechanisms look pretty similar to what I've shown you before. So with all that as a background, these two molecules, and there are soon to be more, are currently in clinical trials in patients with relapsed refractory AML, one from a company, Syndax, that we've been working with. Another company, Cure Oncology, has this small molecule in clinical trials. They're about midway to 3/4 of the way through their phase 1 trials, both of them. They started around the same time, and they've both reported on some of the initial patients. And this is the most recent Syndax reporting from a few weeks ago. And of 31 patients that have been treated with a single agent menin inhibitor in patients that have relapsed or refractory to multiple different types of therapy, 15 of 31 of those patients have had a significant response, such to be called a CR, complete response, but in many cases not complete count recovery. And in 10 of the 15 cases, those patients are MRD negative. That is, by the most sensitive ways of trying to find the leukemia in the bone marrow, it couldn't be found. So about a 54% response rate in MLL rearranged AMLs, and about a 30%, you can see the numbers are small, so these percentages obviously have big error bars, response rate in mutant NPM1. For phase 1s, this is actually pretty good activity and has generated a fair amount of excitement in the field, and we'll see where that continues to go. This is the report from the Cure Oncology trial. Fewer patients, you can see here in phase 1, increasing the dose. And to point out, 2 of the patients at this 200 milligram dose had NPM1 mutations. One of the patients had 7 rounds of different types of chemo to which they were unresponsive. And this patient went into a complete remission with MRD negative, meaning no leukemia that could be found in the bone marrow. So these are really nice responses. Again, small numbers of patients. We'll see how this works out, but at least for the early stages, it looks pretty interesting. So to summarize, I've told you that in a lot of detail that the MLL complex is important in different subtypes of AML. And the 2 subtypes that I've told you about actually account for about 30 to 40% of all AMLs. And we're continuing to look to see if even more might be responsive. And these concepts are now, I don't have time to tell you about this, moving into sarcoma, where other fusion proteins may also be using similar mechanisms, probably different complexes, but similar mechanisms. And these are, as some of you may know, pediatric sarcomas against which new therapies are desperately needed. So I will stop there. This is the lab pre-pandemic, as you can tell. This is our view of Boston from the lab. Tremendous group of people to work with, some of our collaborators here, and some of our funding agencies and collaborators here as well. So I think that I will stop there and I guess move to the Q&A part.
Yeah, so that was wonderful. I really, I think we've learned a lot, and I've really enjoyed the whole spectrum of these findings. We have a couple of questions, quite a few actually, and so maybe I'll just begin in order of the posting. So first question is from Dr. Sonia Selok, who asks, for AML driven by given MLL fusion, do you think that ontogeny and or cell of origin can influence therapeutic vulnerability?
Yeah, it's a good question, and we have studied this in both in mouse models and in using human cells as cells of origin. And it does appear that if the earlier the progenitor, all the way back to just after the stem cell, the more chemo-resistant and aggressive the disease. That is, the earlier, if you transform an early progenitor, that gives you a leukemia that is much less sensitive to either chemotherapy or some targeted therapies. So the answer is yes, the cell of origin can influence the responsiveness. And where we've been able to look at that in clinical trials, it seems to hold up as well.
Yeah, terrific. So we have questions from Dr. Kolya Eppert. So first, question one, menin inhibitor eliminate MLL fusion leukemic cells by differentiation. Does this completely eliminate all fusion cells, or are there some subtypes left behind cells in different states, such as quiescent, perhaps not terminally differentiated? Does the inhibitor need to be taken continuously in a mouse experiment to prevent relapse, or will leukemia return after withdrawing of the menin inhibitor? Some NPM1 mutants may also be implicated. So,
yeah, that's a good question. And so, true, the primary response or primary phenotypic change is differentiation. We can see that both in mouse models and in patients. That is, in some cases, you can look at the patient's neutrophils after treatment with the inhibitor, and you can see the translocation if you're looking at MLL. So the answer is yes, there are differentiated, presumably differentiated leukemia cells in the patient. In the PDX experiments, we only treated for a month or two, and that was sufficient to prevent the leukemia from coming back. Obviously, in humans, we haven't been able to do that yet. So it does seem like prolonged treatment's necessary, but maybe not continuous treatment.
Excellent. So there's a question from Brian Willem. Do you think that chromatin state of the cell of origin influences the course of the disease, or it is simply a reflection of the competency of the cell to undergo a leukemic transformation?
Yeah, it's I think that it's more the former. Chromatin state influences, the chromatin state of the cell of origin has an influence on the ultimate state or phenotype of the leukemia, because we've shown that you can take two different cell types, transform them. They give leukemias that actually don't look too dissimilar. They do have differences, but their chemotherapeutic response is different. So why do I think it's chromatin state? Because we know that there are certain loci that the MLL fusion can bind to in one cell type, but not in the other cell type. So that would suggest that there's something about access to chromatin that the fusion needs in order to activate gene expression. So I do think the chromatin state is influencing the ultimate phenotype, not just the transformation potential.
Yeah, I have another question actually from myself. I'm very curious whether or not what is known about, for example, the impact of MLL, or if there is any impact on non-cell autonomous effects in leukemia, because we know that there's numerous cell-to-cell interactions. There's even equilibria in DLBO that have been described and based on, stem versus non-stem cell interactions and so on. So is MLL or NPM1, one of these genes that triggers some of these non-cell autonomous effects, or is there anything known about that? And is it
so certainly effects on the bone marrow microenvironment have been studied, but not, at least that I know of, specifically in one genotype versus another. That is, in these mouse models, a number of groups have taken the MLL fusion leukemias and watched, if you will, how they remodel the bone marrow microenvironment. They have a dramatic effect on the microenvironment. But I don't know if one genotype is better or worse at doing that than another. I don't think that, at least again, that I know of, has been looked at so closely.
Well, thank you. So there's a question along the same line from Jean-Sébastien Delille, who is asking, thus far, epigenetic therapy has focused on the malignant cell. What are your thoughts on the potential of these therapies to modify the function and fates of other cell types that can impact therapeutic outcomes, such as immune or stromal cells?
Yeah, well, that's a multi-million dollar question. So most pharmaceutical companies and biotechs are trying to figure that out in the immune therapy space for sure. And there have been multiple publications showing that different epigenetic modifying proteins can influence the immunotherapeutic response, be it by influencing the tumor or the immune cells, either one. The problem is it actually seems relatively easy to find that in model systems. It has not been easy yet, at least, to take that and show that it's efficacious in people. So I think the answer is yes, but no one that I know of anyway has really figured out how to use that therapeutically yet.
Okay, maybe I'll try one more question. We have a long list, luckily, but we are short of time. So I'll just put in one more question from Dr. Sonia Sellers. Do you think that ontogeny influences MLL complex composition in infant versus later onset AML?
That's a good question. I don't know the answer to that. It's not a simple question to answer because the biochemistry is not usually done in cell types like those, mostly because of the fact that it's hard to get enough cells. But it's very possible. And as the question kind of alludes to, there are some differences in response and phenotype in adult versus pediatric leukemias that have MLL rearrangements. So that's an interesting question, but I don't know the answer.
Wonderful. So thank you so very much. I think I'm afraid we are out of time. So maybe we could close this session. I'd like to thank you once again for a wonderful presentation. I'd like to then introduce Mr. Barry Cole, who is the chairman of and president of the Cole Foundation, who is ultimately a sponsor and spiritus movens of this event and many other events around the city. So on that note, I'd like to pass the microphone to Mr. Cole. Barry.
Thank you, Yanis. Scott Armstrong, we thank you very much for your stimulating discussion. We try to encourage more dialogue in our community, and you have been a leader today in developing many more ideas that we haven't yet solved, but at least we're now working on the pathway to solve them. So thank you very much for your address. Thank you very much to your assistant, Jessica Brady, who's helped us in arranging this. It's been very good. We thank Amy Ryan from McGill and the McGill team of Sarah Odell, Rebecca McEwen, Jonathan Waugh, Kimberly John, who put this webinar together technically for us. And on behalf of the board of the Cole Foundation, Nancy Wells and myself, we want to thank everybody for participating in this webinar and hope that it stimulates you to develop new ideas to combat the ideas present in pediatric and young adult leukemia. So thank you very much, everybody. And we're sorry that we're virtual this year. Next year, we plan to be in person, so we welcome you live next year.
Let's hope so.
Let's hope so. So thank you very much, Scott. Thank you very much, Yanis. We appreciate your expertise and your development of new ideas.
Thank you.
Thank you, everybody. Goodbye.